I just want to mention that I'm a postdoc in Henry Lester's lab. So we focus on neurobiology, and we have a split between people in our lab that are focusing on nicotine addiction and people that are focusing on Parkinson's disease. And I am one of the people that is focusing on the addiction to nicotine. So, okay. Click. Okay, so because there's been talks about nicotine addiction, a lot of the relevant background's already been provided. So I'll just start by saying that just in the brain, there's many different types of nicotinic receptors. So the two prominent subtypes being the alpha-4 beta-2 and the alpha-7. But I focus mostly on a type that is selectively expressed in midbrain dopaminergic neurons. And so Dr. Hurd mentioned before the importance of the accumbens and the striatum and reinforcement of addiction and initiation of addiction. So these receptors are selectively expressed in these regions. And so I won't go too much detail since there's been relevant background provided already, but I will mention that nicotinic receptors are pentameric. They're ligand-gated ion channels. There's different types of assemblies, and these assemblies can change with drug treatment. So that'll be important a little bit later in the talk. Okay. So what connects nicotinic receptors to the addiction to nicotine? So one of the earliest observations connecting nicotine to nicotinic receptors was the fact that these receptors upregulate following chronic treatment with nicotine. This happens in humans, happens in rodents, and happens in cell lines, cultured neurons. And also, if you individually delete beta-2, alpha-6, or alpha-4 nicotinic receptor subunits, you can prevent self-administration from nicotine ever occurring. And if you selectively re-express these subunits that have been deleted into the ventral tegmental area, you can restore self-administration. And it's important to mention that if you do that in other regions, you will not get self-administration. So in our lab, we've developed nicotinic receptor subunits in rodents that can be selectively activated with lower concentrations of nicotine. So if you activate alpha-4 nicotinic receptors or alpha-6 nicotinic receptors by themselves, that's enough to initiate condition-placed preference. So together, these support the fact that these receptors are playing a major role in the addiction to nicotine, and it's likely that these receptors that are mediating addiction are beta-2 containing, alpha-6 containing, and alpha-4 containing receptors. Okay. So as I mentioned, one of the earliest observations of the addiction to nicotine is that there's an upregulation of receptors. So in essence, that means there's more receptors on the surface of neurons. And so the first observations was made in the early 80s and early 90s, but since then there's been a lot of people contributing to that, or to this story. So what we now know is that nicotine enters into cells. It enters into intracellular organelles. And after this process, it increases the amount of receptors in the endoplasmic reticulum, which is basically where proteins are synthesized, they're assembled, and they're chaperoned out into the surface. So since there's more receptors in the endoplasmic reticulum, there's more that are sent through the secretory pathway, and this results in more receptors on the surface. Okay, we'll skip that there. Okay. So Dr. Hickman went through some of these statistics already, but some other things that are connecting the menthol to nicotinic receptors, which is what I'm working on now, is the fact that recently it's been found that menthol acts as a negative allosteric modulator, so think antagonist, of nicotinic receptors for both the alpha-4 beta-2 type and the alpha-7 type. What's been really interesting for me is that recently, Arthur Brody published a study showing that when you compare people that smoke menthol cigarettes to regular cigarettes, you have more upregulation in the brain. And so that led me to ask the question, what does menthol do by itself? Okay, so one of the ways I did that is just by using mice that we have in our lab that express fluorescent nicotinic receptor subunits. So here we have an example of a mouse where the alpha-6 subunit is tagged to a fluorescent protein called green fluorescent protein, so GFP. So here we have a sample montage of a slice that's cut from the mouse brain, showing the neurons in the ventral tegmental area and the substantia nigra pars compacta, so VTA and SNC. So I can take these mice and treat them with menthol or nicotine, in this case menthol, and I'm using the same treatment paradigm that you saw with Dr. Karkova, so mini osmotic pumps, and I'm delivering menthol at 1 mg per kilogram per hour in this model. And so both in the ventral tegmental area and the substantia nigra pars compacta, I saw a significant increase in nicotinic receptor number of the alpha-6 type with menthol by itself. So I did a similar study using a different type of mouse where the alpha-4 nicotinic receptor subunit is tagged to a different fluorescent protein called M-cherry. And here we're looking at the VTA, the SNC, and the SNR. And so with the VTA and the SNC, we also see a significant increase in receptor number for the alpha-4 type, and no significant increase in the substantia nigra pars reticulata, so labeled SNR. So this is interesting because we've shown previously that nicotine does the reverse, that you have a higher increase in the SNR and a modest increase in the VTA and SNC, but it seems that menthol has a different trend. So the problem with our mouse study is that we can look at increasing receptor number, but we can't tell if that's actually on the surface of neurons or cells. So one way of addressing that is combining two techniques. So one being a technique called total internal reflection microscopy, or TIRF, which is displayed on the right. So typical fluorescent microscopes will image the entire sample, but TIRF uses a technology where you can reflect the laser at a critical angle so that it only illuminates the sample close to the surface. We also have a pH-sensitive GFP analog that's only fluorescent at pH values greater than 7. So what happens when you look at the structure of a cell or a neuron, most of the intracellular organelles are at pH values less than 7. So that means you're only imaging the receptors that are in the ER or in the plasma membrane. So that means in this case we can culture neurons, or you can have cells that you can transfect. So in this case we like to use cultured neurons. So you perfuse with an acidic solution, and what happens is that the fluorescence on the surface is quenched. So that way you can identify the fluorescence just on the ER. That allows you to look selectively at the plasma membrane, and you can look at nicotinic receptor number just at the surface. So this is an example of this right here, and I'll just cut right to the data. So this is a way where I can easily look at combining nicotine with menthol and seeing what happens when you do nicotine by itself and menthol combined with nicotine, looking at specific nicotinic receptors. So when we look at an alpha-4 beta-2 type, we can see that nicotine, as usual, increases receptor number on the surface. Menthol by itself increases, not as much as menthol, but combined, you have an additive effect. And it's interesting because this effect is 25, so menthol with nicotine is about 25% more than menthol by itself, and that's the exact ratio of change that Arthur Brody saw in humans. So I thought that was a good sign that I was on the right track. So when you look at the alpha-6 type of nicotinic receptor, menthol by itself increases receptor number on the surface just as much as nicotine does. But when combined, shows less increase in receptor number on the surface. So another interest I had that in other than just increase in receptor number, what happens to the function? What happens to the assembly of these receptors? Well, it's important to mention, so I mentioned that these receptors are pentamers, and they can exist in different assemblies, what we call stoichiometries. So the most important, or the most prominent type in the brain, the alpha-4 beta-2 nicotinic receptor, which has been characterized the most in the past few decades, exists in two different types of assemblies, one being the high sensitivity and low sensitivity. And that's just saying that they're activated by nicotine at different concentrations. So typically you have a mixed population between these two. So if you use an agonist like acetylcholine, which is the endogenous agonist of nicotinic receptors, or nicotine, you can see that over a range of concentrations you have a population that's activated at low concentrations and another at higher concentrations, so hence the name high and low sensitivity. So nicotine typically alters the assembly so that you have more high sensitivity nicotinic receptors. There being that you have more receptors on the surface that are activated by low concentrations of nicotine. And so using these type of studies where you can expose a range of concentrations, you can look at how these assemblies are changing. So I repeated these experiments comparing chronic menthol treatments to see how the assembly of these receptors were changing. So first looking at alpha-4 beta-2 receptors, so some baseline measurements. So here I'm looking at 0.5 micromolar concentrations of menthol for a 24-hour period. So there's no change in peak current amplitude, so flux through the channel. So if you do longer applications, you look at desensitization because these channels, they open, they activate, and they shut down very quickly, and we call that a desensitized state. So longer applications measure the desensitization. Menthol does not seem to change that, but when you look at the concentration response curve, the typical high sensitivity component that you see with no drug treatment is abolished with chronic menthol treatment. So this is suggesting that menthol is changing the assemblies of these receptors so that you have a low sensitivity form and you lose the high sensitivity form. So when we look at the alpha-6 type of nicotinic receptor, you do see a change in the peak current, but I'll explain why that may happen in a little bit. So no change in decay kinetics. You may think that this is a change, but there's a fast and slow component. So when you do the analysis, there is no change. Okay, so typically when you look at alpha-6, beta-2, beta-3 receptors, they have one monophasic curve, and that's because whenever you have an alpha-6, you usually always have the beta-3 assembled with it together. So, but when you add menthol, you seem to have two different components. The first component aligns almost perfectly with this alpha-6, beta-2, beta-3 component. So the second low sensitivity component at first was kind of a mystery until we figure out a way of looking at just alpha-6, beta-2 receptors without the beta-3, and that aligns perfectly with this low sensitivity component. So suggesting that the typical alpha-6, beta-2, beta-3 of these receptors is modulated so that menthol can assemble these receptors and place them on the surface without a beta-3 subunit. Okay. So this is just some preliminary data. So recording from slices. So right now I'm working on figuring out how menthol is affecting VTA dopaminergic neurons. So preliminarily, the baseline firing rates does not change with menthol, and it seems like peak current amplitude does not change, but there is a change in the decay kinetics, so basically how fast they deactivate, as summarized here. And that may be an indication of change in assembly, but that's also what some of my other studies have been showing. So I'll just say that I've done FRET analysis, and this just supports the fact that the assembly of these receptors is changing following menthol treatment. Okay, just to summarize, so menthol by itself does increase receptor number. It does this in the intracellular organelles, it does this on the surface. So at low concentrations, or the concentrations that I've examined, menthol seems not to change the functional properties of the receptors, but there needs to be a lot more studies looking at things like, how the channels are opening, how fast and how often. But it does show that menthol is altering the assemblies of these receptors. And this is just, that's just a cartoon of what the summary is. Okay, so I'd like to thank my advisor, Henry Lester, the members of the Lester Lab. So NIDA for funding, as Dr. Avila mentioned, so I'm a postdoctoral fellow of NIDA, and he was my program officer, so all thanks to him. And thanks to Caltech for their support.
Very nice talk, and it's very interesting data. In terms of localization of nicotine in ER, do you see what type of ER, like it's a smooth ER or rough ER, and how you localize? Did you do any immunogold electron microscopy to localize this?
So far what we've done is mostly with the peripheral ER, just with the limit of what we can image. So my advisor is actually on sabbatical specifically trying to answer that question by developing nicotine sensors. So there's been a lot of data suggesting that nicotine gets into the ER, but no real physical measurements. So we've been able to fluorescently label agonists and do some level of fret, but really when it comes down to it, that's not a very sensitive measurement. So he's working on that problem right now.
You know, this is very interesting. Our group is also, interested in nicotine and cell death. And this ER, it could have directly induced ER stress, and which in turn trigger cell death. Not only neurons, any other tissues also. Thank you. It's very nice talk.
Well, if you're interested in cell death, you should definitely talk to Henry because that's kind of an extra arm of our research wing that we previously were working on and looking specifically at dopaminergic neurons, how low concentrations of nicotine, so 100 nanomolar or less, preserves the neurons even in cases where you're stressing them.
So can you talk about any clinical relevance of this? If someone uses menthol, how does that affect nicotine binding and things like that?
So I have a lot of other data that I didn't show here. So it seems like menthol increases the equilibrium binding time. So like the time it takes for nicotine to bind and stabilize the receptor, it seems that menthol it reduces the time, so it makes it more efficient for nicotine binding. As far as like other clinical implications right now, so I'm in the process of doing behavior work and looking at how menthol affects behavior. That's very preliminary, and for those of you that work with mice, you know that there's a lot of variability, so you have to have enough data before you say anything. Because I don't want to say, this is what it is, and then you see me at SFN and be like, that didn't happen. But if you are going to SFN, by then I'm 99% sure it'll be done, so I can talk about it at SFN. So
when you look at circuitry, have you looked at other brain areas?
Not yet. As far as the imaging, so I've started with the imaging because that's a lot faster than, say, slice physiology. That takes a lot more time. So what the goal is, so we've developed a pretty good circuit analysis over the years with nicotine effects just through electrophysiology about how the upregulation and GABAergic neurons in the SNR, how that affects the dopaminergic neurons in the VTA and so on and so forth. But since right now I'm the only one working on this menthol project, so that's going to take, if we're going to take that approach, it's going to take years and other people. So.
Okay, great. Thank you, Brandon. And now we have